Is active learning enough? A large-scale quasi-experimental study of the role of misconceptions in evolution learning outcomes. Evolutionary misconceptions have been discussed for at least a century, and a large literature has described and cataloged them. Misconceptions or naive ideas is a term that has changed in meaning over time and been used to encompass many different types of ideation, including universally false beliefs, normative ideas that become false beliefs as a result of overextension, and a mixture of normative ideas and false beliefs. In recent years, significant attention is focused on the role of active learning because it's been shown to be important for learning in a variety of fields. However, the role of active learning in evolution instruction specifically remains ambiguous. In addition, there's remarkably little work testing whether devoting instructional time to explicit discussions of evolution misconceptions is beneficial, neutral, or harmful for evolution learning. Often, instructional materials and interventions are designed to foster normative scientific models, and in doing so, address misconceptions implicitly. Of course, this doesn't mean that instructors have not been teaching misconceptions explicitly, but no work indicates the extent to which educators are using this approach. Despite a century of discussion about evolution misconceptions, the amount of life science education explicitly devoted to them remains unknown. Overall, insufficient empirical research has been devoted to the roles that both misconception-focused instruction and active learning play in evolution learning. We address this gap in the literature by asking the following research questions. Is active learning associated with learning evolution? Is misconception-focused instruction associated with learning evolution? And does misconception-focused instruction explain evolution learning above and beyond active learning alone? We designed our study to address the common limitations of other studies on evolution learning. We use robust measures of active learning and time spent in misconceptions instead of relying on self-reports. We also use robust measures of evolution knowledge using validated assessment instruments. We controlled for evolution acceptance, also using robust measures of this construct. And the course that was the focus of this study was led by a typical biologist with no formal education, training, or DBER experience. We measured evolution learning at multiple time points within each semester and controlled for demographic and background variables. The data for this study were collected from 4 semesters of an introductory biology course that took place in a lecture hall style room that seated about 250 students. Over 400 students completed the surveys in each semester. The course content is aligned with the 5 core concepts of biological literacy outlined in Vision and Change. Evolution was taught in the first half of the semester by a biologist instructor with no formal preparation or training in biology education or in active learning. 4 iterations of the course were studied with differing amounts of active learning and instruction time devoted to misconceptions. This table outlines the measures used for this study. To measure active learning, we used the COPUS instrument, a validated classroom observational tool that documents active learning-aligned behaviors. To measure time devoted to misconceptions, we documented the total time spent on misconception-focused activities during class. We measured evolution knowledge using 3 validated and published assessment instruments, the SINs, the CANs, and the ACORNs. Evolution acceptance was measured using the IC instrument, and race, gender, and prior biology experience were reported by the student at the beginning of the semester. This table shows additional detail on the evolution knowledge instruments used in this study. The SINs and the CANs are closed response surveys with 4 to 5 answer options, one of which is correct. Misconceptions are incorporated in the survey as item distractors. And these surveys test whether a student prefers a normative or scientific idea over a misconception in multiple trait taxa contexts. The score is reported as the sum of correct responses. The ACORNs is an open response item scored for key concept and naive ideas. The naive ideas are the misconceptions. The item tests for the presence of key concepts and naive ideas in multiple trait taxa trait polarity contexts. Separate scores for scientific ideas and misconceptions are provided, and comparisons across items, such as a plant item and an animal item, generates measures of model consistency. Students' written answers to the ACORNS questions were uploaded into the Evo Grader scoring software. This figure shows the study design. Each color represents one of the 4 course iterations. Evolution knowledge data were collected from participants at 3 time points: at the beginning of the course, after the evolution unit, and at the end of the course. The iterations range from least evidence-based instruction to most evidence-based instruction, as indicated by the arrows shown here. In the least evidence-based iteration, there was no explicit focus on misconceptions. The remaining 3 iterations all included a misconception treatment as indicated by the capital T. This treatment occurred between the pre and the post-test. The nature of the misconception treatment differed across iterations as shown on the right. However, I won't go into much detail here, and instead I'll focus on the time spent in misconceptions and in active learning, both of which increased throughout the iterations of the course, as you can see on the right side of the screen. These colors will remain consistent throughout the remainder of the talk. We analyzed these data using multi-level linear and logistic regression models. For each evolution learning outcome variable, 3 models were run which differed in their predictor variables. One included the percent active learning, one included the percent misconception time, and a third model included both of these variables. In the following slides, I'll present results for each of the 4 evolution learning outcome measures. A figure will be shown for each outcome measure, and it will look something like this. The key on the right of the figure indicates that the black line represents the iteration with the lowest amount of evidence-based instruction, and the blue line represents the iteration with the highest amount of evidence-based instruction. For the SINs, there was a significant and positive relationship between evolution learning and percent active learning. There was also a significant and positive relationship between evolution learning and percent misconception time. When both variables were included in the model, only percent misconception time remained significant and positive. The same general pattern was found for ACORNS key concepts for both the plant and animal ACORNS items. A similar but opposite pattern was found for ACORNS naive ideas. And finally, for ACORNS model consistency, both percent active learning and percent misconception time were associated with having a consistent scientific model, but only percent misconception time remained significant when both variables were included. In this study, both active learning and misconception-focused instruction were significantly and similarly associated with knowledge of core evolution key concepts, the absence of misconceptions, and the consistency in evolutionary reasoning across contexts. Furthermore, misconception-focused instruction explained each of these outcomes above and beyond active learning alone. Although the impacts of misconception-focused instruction on evolution learning outcomes have not been the subject of much research, many active learning interventions may indeed incorporate this instructional strategy. Indeed, in this study, the amounts of active learning and misconception-focused instruction were correlated in our sample, with 36 to 66% of misconception-focused instruction including active learning. The historical focus on the benefits of active learning and the inattention given to misconception-focused instructional strategies may offer an incomplete picture of what classroom features best support evolution learning. This work suggests that misconception-focused instruction should be utilized in evolution education. At present, no work indicates the extent to which educators are using this approach. It also suggests that active learning alone may not be sufficient for helping students learn evolution. Experimental intervention studies that parse out the relative impacts of active learning and misconception-focused instruction are crucial next steps to understanding how to best support evolution learning in undergraduates. The importance of improving this understanding cannot be overstated. Equity and diversity work suggests that URM students arrive in biology classes with significantly lower knowledge and greater misconceptions of evolution. Using effective approaches for helping students learn evolution is central to addressing these disparities. We'd like to thank our funders and Dr. John True for his assistance.